Mm-mm. I didn't. I wasn't talking to you. Body on body. There is no interruption in the metabolism. I'm just gonna. Okay, the goal is to speed read through all of this, and then I'll go over it with a critical eye. After I go over with a critical eye, then I'll try to understand it. So I'll read the article 2 times over. So that's gonna be 40 pages. No, that's more than 40, like 52. Nah, my math is wrong. Anyways, there's no interruption in metabolism. Metabolism. God, dude, stop. Giant. I'm trying to understand this, bro. Bro. There's no interruption in metabolism. Bol. Okay, the cell remains coordinated as they had been, and their gene expression and potential remain equivalent to what they had been. If embryos split at the four cell stage, for example, all the cells before and after the split would be to- oh my god, bro. Topia refers to a type of cell that has the ability to divide and produce all differentiated cells. Example, zygote spores, so its ability to divide. Okay, would be potent. Further, it at the four cell stage, two of the cells were killed. LTG would presumably not find a substance change in the remaining cells. After all, cells die during development, all the time with no ontological consequence for the embryo or fetus. But if two of the cells were removed and then immediately killed, LTG should claim that the original entity was destroyed since two twins of the same size were generated, however briefly, but nothing about that surviving two cell embryo would be different in the two cases. We would have no basis for calling them two different organisms. You're not gonna get me to read. even in a in one context or another. For example, the more demand the given a given instance, a particular change in level of some protein or, or protein modification only affects the probability of a cell taking on even the most radically different developmental path. Sometimes the cell's fate will change, and sometimes it will not, as the efficiency rate for various protocols for inducing pluri- pluripotency attest. In these cases, that could mean the difference between a cell that could generate an entire organism, so totipotent, the dividing cell, or I argue, a pluripotent cell. I need to look these terms up. I'm mispronouncing them badly, but it's fine. I'm just gonna skim through, okay? I understand this kinda, and we cannot are looking for a substantial change or accidental change, even if the distinction is meaningful. The example of the Parthenode, so Parthenode is just a embryo produced through parthenogenesis. Parthenogenesis is whereby the uh, the ovum alone is gonna be the sole basis for the activation of the blah blah blah. I discussed above further show that we cannot see substance changes occur in development, even the cases where strict ontology requires that substance changes take place. Obviously, in the case of paranoes, for the egg to retain its chromosomes would not be a substance change. It's not. All changes simply divided should not be a substance change according to LTG, since the zygote and the clever stage embryo divide, and LTG see no substance change there. The only other necessary step in parthenogenesis is either the flow of an electric current or the flow of um, calcium ions. Both these very subtle phenomenons occur during many events. Fuck that T would classify as accidental if either substance phenomenon or substance, then it could frequent substance changes in development that we could fail to notice. Since we cannot always tell a change in progress whether it is a substance change, we might hope to rely on retrospective analysis by comparing entities before and after change occurs or happens to take place. Unfortunately, we cannot rely on this criterion either. The dissociation and reassociation of early embryos provide an example of a scenario where strict ontology required us to conclude that multiple substance changes have taken the entity at the beginning. A strict ontologist would view the dissociation of the embryo as the substance change that leads to twin formation. The resonation would be second. I I didn't promise you a proper read through. Okay, so I'm just gonna keep going. There'd be no way to tell what happened. Even if we didn't be watching while the cells separated and drawing, if anything bad happened to the doll. One could argue these are rare events, but complete separation of cells should not be necessary to effect a substance change. If we were to accept that substance change according to these cases, cells in an embryo respond to each other because of signaling. If the signaling between the cells, I understand this part. I like that. In an embryo, were to fall below some threshold and then to exceed that threshold again, that should be as much of a substance change as physical cleaving of the embryo. Changes in signaling levels can be quite rapid. We have no objective criterion for establishing the signaling threshold. We have no way of knowing how often threshold is crossed in either direction, since even when LTG posits the occurrence of a substance change, entities before and after the event can be indistinguishable. We cannot reliably distinguish substances changes even retrospectively by comparing entities at two different times. We cannot therefore interrogate early development, interrog early development, and reliably identify the final substance change. We certainly cannot conclude from introspection that in typical human development, no substance change occur after fertilization. I think I have an idea. Maybe I can put this all in GPT, ask user research question. I'll ask them to give me like YouTube links for all the words I don't understand because I don't study biology, brother. So I'll get them to ask like what is, let's say, let me find a word I don't get. So let's say I don't understand um cleavage state. I just provide me a YouTube link, stuff like that, and I'll be able to organize it in my Obsidian. That'd be good. Okay, shared goals. Observe that anatomy and behavior of the earlier stages in humans develop benefit earlier. Observation that zygotes pursue the same telos or goal as adults. Survival and development of the whole. LTG further concludes that the shared telos would imply that zygotes and adults share the same substance until no substance changes takes place after fertilization in a typical human. I discuss why teleological reasoning should have no place in this discussion of human development in part 3 here. It is sufficient to point out that LTG inference and conclusion do not follow from their observation because even if earlier stages and later stages are different entities, earlier stages indisputably give rise to later stages. Far from being surprised, we should expect to see that entities uh, invest in their posterity in such a way that the structures they build and the behaviors they engage in will benefit later stages. We routinely observe this type of strong investment even when dealing with such indisputable different beings as parents and their offspring. Natural selection is perfectly sufficient to explain such development. Wait, to explain such investment in the context where we observe it. If one takes seriously the possibility of substance change during development, survival, and development may not be compatible goals. It is only if we assume that none of the changes that take place in development have ontological significance that we should necessarily view development through embryonic and fetal stages as futile to telos of a single entity. When LTG takes this development as evidence that no substance change has taken place, they engage in circular reasoning. Mmm, I have another idea. So I can ask it to explain the words by giving YouTube videos short explanations of how it explains in the context. Also, every fallacy that the article points out, such as this one, and explain how these fallacies are the fallacies, you know what I mean? And that way I can understand better. Okay, we're at page 9. Oof. We cannot therefore rely on the fact that the behavior and anatomy of the embryo helps to ensure the development and survival of the adult, is evident that no substance change occur between these developmental stages. Fundamentally different pathways. LTG suggests that we can discern retrospectively where have been substance changes because in those cases the result is fundamentally different developmental pathways, whereas they are not substance changes. The alteration in genetic profiles are more reasonably understood as adaptations by the organisms to their changing environments. We can identify many events in the human development where the embryo embarks on a fundamentally different developmental pathway. If such developmental change is sufficient to identify the substance change, even the face of metabolic and physiological continuity of an organism, Any of these should satisfy LTG's criterion for substance change. Gene expression is a fundamental to the identity and developmental stage of cells and organisms. During cleavage stage and through implantation, global gene expression changes and can indicate that human development trajectory shifts several times. Egg and sperm do not transcribe many genes from their own genomes, even upon fertilization. The zygote relies on the mRNAs, and proteins that have been disposed in the egg prior to ovulation in order to regulate the early embryo's metabolism. I can understand this, yeah. The first cell division. Some human embryonic transcription initiate at two cell stage, with more gene turning on the fourth stage. Later, at the blasto stage, a major wave of transcriptions turn on genes required for an organism to develop. On implantation, we can see many of these genes' expression change reflected in the dramatic remodeling of the genome epigenetic structure. For example, DNA is a pre-implantation stage is hypothetical, hypomethic, these are one of the words, hypomethylated, a state that correlates with totipotency and pluripotency. Again, I don't know these words. I'm just gonna do a quick speed read through, okay? But these are global wave of I don't know that word either. Gosh. Take a mind like break. Okay, I'm probably gonna do a skip through. The reading stuff is not. Oh yeah, I'll read it. I need to stop. My voice hurts. So at the same stage, when we observe that some of these global transitions in epigenetic genes and gene expressions, we also see other changes that might mark transitions to fundamentally different pathways. For example, two cell stage is the first time we see cells in the embryo with a complete diploid genome. Since in the zygote, by the time the maternal and paternal genomes come together, each has replicated on its own. At implantation, we see for the first time the capacity to grow rather than divide without growth. And then the onset of extensive signaling between mother and embryo, without which further development is impossible. If we were to accept that without further development is impossible, if we're, oh my god, for some change are substantial and other accidental, these development transitions and could be seen as substance changes based on gene expressions and changes and developmental trajectories. Many authors, in fact, have pointed to them as substance changes. Why did he cite himself? Look, look beyond, dude. Look beyond, looking beyond changes in gene expression. Several changes in endorin. Oh my god, endor endorin or nervous system later. Don't shift and trajectory depend on the fundamental thing. Pathways. Since can always take and change the question and fertilization looking at our node. So since in this with the adult and then the diagonal tendencies according to Ontology, these are the different development stages could derive the dornus, stem versus organism, LSP, and the zygotes in the entire result only from so once change, development argue that using strict ontology arguments the zygote is no more a person than our other cells. Entire different changes. Shouldn't present the problem for LTG because it's not all clear whether how a substance change could be occurring in IPS generation. All the steps in the process that look just like changes in LTG who would accept as accidental or in other contexts. LTG dispute my argument as I go to an IPS cell, yes, cells should have been the same cell more styles according to that with the IPS or the stem cell.